grapevine plant were treated with compost tea and microbial consortium. In the field, after this treatment, phenotypical responses were expected to be evaluated. This response may include changes in the plant growth, stress tolerance, or resistance, as it was presented in the previous presentation. These phenotypical responses can be driven by biological mechanisms such as priming, post-transcriptional regulation, or physiological and biochemical adjustment. But it can also be caused by a molecular response, which involves change at the gene expression level, which is the focus of our work. So our objective is to analyze gene expression response of grapevine to the microbial consortium 3P and compost tea treatment. This table shows the list of grapevine leaf samples that we received. The first treatment was applied in June, and samples were collected 24 and 48 hours after treatment. For the moment, we are focusing on these samples. In addition, we received also samples that were treated in August. Here I will explain a little bit about the experimental workflow from sample processing to the gene expression analysis. But I should mention that we started this work just a month ago, so we still don't have results. But I will explain what we did already and what we are going to do. So the first step is the extraction of the RNA. We started by grinding the leaf samples in liquid nitrogen, and then RNA extraction is then performed. But it is important to mention that grapevine leaves are rich in polyphenols and other secondary metabolites, and this interferes with the RNA purity and yield, making the RNA extraction a little bit challenging. For this reason, we tested a few different extraction methods. First one, we tested the commercial kit from Qiagen blend, which is the RNeasy plant mini kit, and this kit didn't give any RNA, so we excluded it. Then we tried from the same brand another kit, and this one was supposed to be a little bit better adapted for difficult samples. We were able to extract, but it was mainly genomic DNA rather than RNA, as you can see in this gel. So the thick band is DNA and the thin one is RNA. Finally, we tested the setup method, which is the traditional way, and it worked. We tried a few samples and it worked, so we decided to extract all the samples with this method. Now we are in this step. After RNA extraction, we proceed with RNA quantification and quality control. For this, we use the qubit for RNA concentration and Tecan spectrophotometer for the total nucleic acid and to check the purity of the sample. After that, RNA samples are treated with the DNAs to remove genomic DNA and then reverse transcribed into complementary DNA, which is or cDNA, which represents the expressed part of the gene to be used for the qPCR. Gene expression analysis are then performed using real-time quantitative PCR on the complementary DNA with the gene-specific primers. Then the resulted CT values are normalized using housekeeping genes and compared to the control samples. For gene expression analysis, we selected a few target genes which represent the major grapevine defense and stress-related pathways, which include salicylic acid pathway, jasmonic acid, oxylipin, and ethylene pathway, also phenylpropanoid, phytoalexin pathway, pathogenesis-related protein or the PR genes, and redox regulation. And in addition to the target genes, we selected several reference genes that we included for the normalization of the qPCR. This is the list of the primers that we are using. And this is the curve of that we used to try few primers. So for primer validation, and most of them showed a good result, even with a very low concentration of RNA. At the current stage, we have the extraction protocol for RNA already optimized, and the primer set is already defined and validated. Next, we will continue with the extraction of RNA using setup method, validated the quality and DNA removal, and then go on with the gene expression analysis using the qPCR, then analyzing the data. Thank you for your attention.